Within the sites where the antigen is concentrated, B cells compete with each other for signals and space. This is a very effective way of ensuring that, from all the antibodies available, the most effective are amplified rapidly. Within the germinal center, the B cells therefore undergo carefully coordinated rounds of proliferation. The result of this is a surge in antibody-secreting B cells, which eventually leave the lymph node and can be detected in the blood a week or so after an infection, accompanied by a rapid rise in antiviral antibodies. It is worth noting that not all antibodies directed against viral proteins are equal. Antibodies directed against the viral envelope proteins, which direct the attachment and infection of virus to host cells, are typically of the most protective value, although they are not always successful in neutralizing the target. If the virus is bound and recognized by the immune system in alternative ways, e.g. via complement, this may also provide some protection. Antibodies which bind a virus and prevent it infecting further cells are called neutralizing antibodies. Producing high levels of neutralizing antibodies is the goal of many vaccine strategies, as this can provide complete protection against infection, a so-called sterilizing immunity. The difference between antibodies that bind and those that neutralize became very important during the COVID-19 pandemic. The initial innate response and accompanying interferons, followed by rapidly emerging cellular T cell and humoral B cell/antibody immunity, should reduce the number of productively infected cells and the spread of the virus between cells. This reduction in virus replication is important in the evolution of immunity, since it means a reduced level of antigen and innate triggering. Here, the B cell response and the T cell response diverge somewhat. B cells continue to mature and generate antibodies, and typically, after infection, these antibodies can be detected in the circulation for the rest of that person's life. Such antibodies are produced by B cells which have migrated to the bone marrow and actively secreted antibodies, having now become a specialized cell known as a plasma cell. T cells, in contrast, are, in the main, much more short-lived. The response, as mentioned, expands very rapidly, and effector CD8+ T cells reach very high numbers in the blood and tissues. In the absence of further stimulation, however, these cells die quickly, and the population sharply contracts. This dynamic expansion and contraction makes sense in limiting the exposure of the body to very dangerous killer cells, which always have the potential to cause excessive tissue damage, i.e. immunopathology. However, this population is not lost altogether, persisting long-term as an immunological memory.